me begin this lecture by reminding you where we are in the course. Up to this point, we've explored the concept of genetic information in living systems on the molecular level. We began this discussion, you'll recall, with the fundamental question: how is it possible for offspring to inherit traits from their parents? Now, this discussion first led us to consider the remarkable structure of the molecule DNA, and we looked in detail at how this structure enables DNA to serve as an information-bearing molecule. We then looked at the processes by which the information stored in DNA is transmitted via RNA into the structure of proteins through the processes of transcription and translation. But this discussion of what we'd call molecular genetics has not completely answered the question of how offspring actually inherit their traits from their parents. We know DNA can encode information, and we know this information can be copied and decoded to make proteins, but how does the DNA itself actually get passed from one organism to another? In specific, when a cell divides, how's the DNA in the dividing cell physically distributed to the two resulting daughter cells? More generally, when a new individual is produced, how does it acquire its DNA from its parent? Now, to answer these questions, we need to move our discussion from the molecular level of the hierarchy of biological organization to the level of cells and organisms. There really are two issues we need to start addressing. First, we need to understand how the continuity of genetic information is maintained when cells divide, to ensure that daughter cells have the same genetic material as their parents. Cells normally divide through a process called mitosis, and so we'll first look at mitosis and see how DNA gets distributed in this way. Second, we need to look at one special way in which many kinds of organisms produce offspring, that is, through sexual reproduction. Because this kind of reproduction introduces variation into the genetic makeup of offspring. Sexual reproduction involves a different sort of cell division that we call meiosis. And so then we'll turn our attention to how meiosis differs from mitosis and see how that leads to continuity and variation in the transmission of information across generations. Well, let's begin by looking at how DNA is distributed in normally dividing cells through the process of mitosis. Now, single-celled organisms, of course, simply reproduce by dividing in two. And when they do, each daughter cell has to get a complete copy of the original parent's DNA. In multicellular organisms such as ourselves, cells divide for all sorts of reasons other than reproduction. For example, when we grow, cells are dividing, or sometimes older cells will die and be replaced by younger ones. In these cases, it's equally essential for the daughter cells to have the same DNA as their parents. So how do we get two copies of DNA in a parent cell sorted out into the daughter cells after division? Now, in prokaryotic cells, this isn't such a big deal because, as I told you before, prokaryotic cells have a relatively simple piece of DNA that serves as their information-bearing molecule. It's just one circular piece of DNA, and it forms what we would call a single chromosome. So when a prokaryotic cell, and all of these prokaryotes are single cells, divide, you can imagine it's not too hard to imagine that one copy of this circular chromosome would go to one cell and the other to the other cell. But in eukaryotic cells, there's typically a whole lot more DNA, a whole lot more. And furthermore, this DNA is divided into a number of different linear strands. These linear strands are what we would also call chromosomes, and individual organisms might have many, if not dozens, of these chromosomes. So sorting out chromosomes in the process of division for eukaryotic cells is potentially a much more complicated problem. Now, what I want to do is to begin this discussion by turning our attention to the life of the cell, or what we'd call the cell cycle. The cell cycle can be divided into two major phases. In the mitotic phase of the cell cycle, that's when the cell actually divides, splitting in two to form two daughter cells. Interphase refers to the remainder of the cell cycle. Interphase is typically when the cell does what it normally does in life, and it's much longer than the mitotic phase, occupying up to about 90% of the entire cell cycle. Now, there are a lot of details to the cell cycle that we could discuss. I mean, for example, interphase is typically divided into its own three phases, the so-called G1 phase, the S phase, and the G2 phase. These stand for the first gap phase, the synthesis phase, and the second gap phase. The only point I want to make here is that during the S phase of interphase, S phase for synthesis, that's when the DNA in the cell is being replicated. That's when that process of DNA replication that we discussed a number of lectures ago actually is occurring. Now, once the DNA has replicated, the cell may go into the mitotic phase where it divides. And what we have to do is to get these two copies of all of the DNA separated. Let's take a closer look then at mitosis and see how this happens. Mitosis is really an extraordinarily beautiful process. If you look at mitotic cells under the microscope, you can almost see this remarkable choreography of the movements of chromosomes. Now, normally during the interphase of the cell, the chromosomes in the cell's nucleus are dispersed. You can't actually see them under a normal light microscope. But when the cell prepares for mitosis, these chromosomes, and remember, chromosomes are made up of both DNA and proteins associated together, a structure that we call chromatin. These chromosomes begin to curl up on each other and condense and form little physical bodies that you can actually see under a normal low-powered microscope. Now, for our purposes, there are 4 stages of mitosis to consider, which we define based on the progress of the movements of chromosomes in these dividing cells. Of course, mitosis is also a complex cellular process, and there are a lot of details that we could look at here, but I don't want you to get lost in the details. Instead, I want you to focus on where the chromosomes are going, how they're moving. The first stage of mitosis is what we call prophase. Prophase occurs when the chromosomes in the nucleus first start to appear, first start to condense. The other thing that happens during prophase is actually the nucleus begins to break down. The membrane that surrounds the nucleus begins to dissipate, and it disappears if you're looking at it under a microscope. Now remember, at this phase, each chromosome actually consists of 2 copies. Now I'm going to give you some terms, and I want you to try to sort these out because it'll be important to keep these in mind as we talk not only about mitosis, but later about meiosis. The 2 copies of each chromosome are physically attached. They remain attached at a little structure somewhere in the middle of the chromosome that we call the centromere. And those 2 copies are what we call sister chromatids. So if you look at a chromosome under a microscope, you typically see sort of 2 parallel lines that are connected somewhere in the middle. And these 2 parallel lines really are the 2 copies of that chromosome that were made during the synthesis phase and interphase. What mitosis will do is separate those 2 copies, separate those sister chromatids with 1 chromatid going into each of the daughter cells following division. Now, the second major waypoint, if you will, during mitosis is what we call metaphase. Metaphase is recognized by the fact that all of the chromosomes that have condensed in the cell physically line up. I mean, this is where the ballet comes in. It's not like they're doing a little square dance. They're lining up right along the center of the cell, really in a plane in the center of the cell. Now, at this time, these chromosomes are actually attached by very small protein structures that are called spindles. These are structural proteins that connect back to little structures, protein structures, again called centrioles, at either side of the cell. So it's as though each chromosome lined up in the middle of the cell has a little wire or string connected to it, leading back to an object on either side. What happens next is what we call anaphase. Anaphase is recognized by the fact that the chromosomes actually pull apart. Now, what really is happening is that the 2 sister chromatids are being separated. The centriole comes apart, and the spindles will literally pull the 2 sister chromatids away from each other for each chromosome, pulling 1 chromatid to one side of the cell and the other chromatid to the other side of the cell. And then finally, the final phase of mitosis that we want to label is called telophase. Telophase is when the parent cell actually divides. It's as though there's a drawstring that was positioned right around the center of the cell in the same plane where those chromosomes had lined up during metaphase. And after the chromatids had separated, that drawstring gets to be pulled and starts squeezing the center of the cell until the cell literally divides into what are now two daughter cells. And the important thing to keep in mind then is that what we've accomplished is the separation of the chromatids, the copies that were made earlier in interphase, into the two sides of the cell. And that's how we can account for the continuity of information as cells divide. Those copies are choreographed to go to each of the two daughter cells. Now, actually, mitosis isn't really too hard to visualize. If you just think of each chromosome having copies and those copies lining up such that they get pulled to each side, then it's pretty clear how we can get the DNA that it was copied earlier to be segregated into daughter cells. But it's important to keep those basic chromosome movements in mind, because they'll become even more important for us to understand as we next look at the process of meiosis. Well, why do we care about meiosis? This has to do with sexual reproduction. If organisms only reproduced by simply dividing cells, then mitosis would be all we need to understand to explain how genetic information is transmitted across generations. But many organisms, especially multicellular organisms such as ourselves, produce offspring not simply by dividing cells, but by combining two cells to form a single cell. Now, that's why we need to understand meiosis, because it's the cells that are combined, what we call gametes, that need to be produced in a special way. Before we talk about that special way, let me remind you, though, what's really happening in sexual reproduction is that by combining cells from two different parents, we're combining genetic information from those two individuals. And thus, the genetic makeup of the offspring is a combination of the genetic makeup of the two parents. Understanding the movements of chromosomes in meiosis becomes essential then for understanding how traits are transmitted from parents to offspring in sexually reproducing organisms, something that we'll call in the next lecture Mendelian genetics. Now, a couple of terms here. We say that if an organism has two copies of its genetic information, then it's called diploid. Two copies, diploid. Sexually reproducing organisms typically are composed of diploid cells. Why is that? Because they get a copy of their genetic information from one copy of their genetic information from one parent and another copy from the other parent. So, for example, each of us in our own cells has 23 different kinds of chromosomes, with each chromosome type having its own set of genetic material, having what we now know to be its own set of genes. But each of your cells has actually two copies of each of these chromosomes. So if you just looked at the number of chromosomes total in your cells, you'd count 46. But if you look at them closely, and you can do this under a microscope by looking at their morphology, you see that these 46 chromosomes really represent 23 pairs of the same kind of chromosome. Remember, one version of each of these pairs has come from your mother, and one version has come from your father. Now, pairs of the same kind of chromosome in a diploid cell are called homologous, or homologous pairs, or I might refer to those two chromosomes in a pair as being homologs. Different kinds of organisms have different numbers of chromosomes. For example, if you own a cat, your cat has 38 chromosomes total. But these 38 chromosomes, again, represent 19 homologous pairs. And the cat's received one set of these chromosomes, 19 of them from its mother, and the other set, 19, from its father. Now, this is kind of interesting, because when you think of this in terms of cell division, it presents a particular kind of problem. If each offspring gets a set of chromosomes from its father and another set of chromosomes from its mother, then why don't offspring continually have more and more copies of their chromosomes? So, for example, in a diploid organism, the mother and the father both have 2 sets of each chromosome. If they produce cells that will combine to form another offspring, why doesn't that offspring now have 4 copies of each chromosome? The answer to this comes from meiosis, which is the fact that sexually reproducing organisms, when they undergo meiotic cell division, actually reduce by half the number of chromosomes they put into their cells, the cells that are produced by this. And they do so in a way that actually keeps 1 copy of each chromosome type in the newly created cells. The cells that are created through meiosis are what we call gametes. So, for example, sperm and eggs are 2 different kinds of gametes. Gametes are haploid cells. And by haploid, I mean they only have 1 copy of each chromosome type. Now, this confused me when I was a student. We say diploid cells have 2 copies of each chromosome type. Why don't we call these uniploid cells? I don't know. This is the way biologists worked back then. Anyway, haploid cells are considered to have just 1 copy of each chromosome type. Meiosis is the way that cells achieve the production of these haploid gametes. So let's look at meiosis now in a little more detail. Meiosis is similar to mitosis in some ways. As was the case for mitosis, the DNA in a cell has already been divided, has already been replicated, I should say, before the process of meiosis begins. In other words, again, in the synthesis phase of interphase, the cell has actually duplicated every one of its molecules of DNA, every one of its chromosomes. And as these chromosomes begin to condense, again we see that every chromosome actually is composed of 2 sister chromatids. The 2 sister chromatids are the copies of the DNA that were made earlier during the synthesis phase. But there's a fundamental difference between the way meiosis and mitosis work. And this is the key. There are actually 2 rounds of cell division that occur in meiosis, not just 1 round. The first round of cell division actually separates the homologous pairs of chromosome into the daughter cells, creating haploid cells from what were diploid cells. But these chromosomes that are separated after the first meiotic division still each are made of 2 copies. They're still 2 sister chromatids attached together. That's the subtle part. Remember, you've got 2 of each kind of chromosome, 1 from your mother and 1 from your father. When you go through DNA synthesis, all 46 of your chromosomes, all 23 homologous pairs, each are replicated. So now you have 46 physical chromosomes, each of which has 2 copies, which we call sister chromatids. But each of those 46 chromosomes can be assigned to 1 of 23 homologous pairs. So when we're going into meiosis, think of each chromosome type as having 2 versions, 1 from the mother, 1 from the father, and then each of those chromosomes themselves being 2 copies connected together. Okay, so what happens then in the second meiotic division is after we've separated the homologous pairs, then the second division will separate the sister chromatids. Now, let me make that clear by looking at the stages of meiosis in a little more detail. I'm going to refer to these 2 cell divisions in a variety of ways. I might refer to the first meiotic division, or simply meiosis 1, versus the second meiotic division, or more simply meiosis 2. Let's look at the first cell division that occurs in meiosis. Now, the first stage is again what we would call prophase, but we're going to call this prophase 1 now because it's the prophase of the first meiotic division. And in prophase 1, as we saw with mitosis, the chromosomes have condensed and the nuclear membrane breaks down. But unlike mitosis, And this is the key thing to keep in mind. What happens is that these homologous pairs begin to associate very closely. In other words, the two homologs that are members of a homologous pair actually come together and are adjacent to each other during prophase. This didn't happen in prophase of mitosis. All the homologs were on their own, but in meiosis, in the first prophase, the homologs actually associate. Interestingly, during this association, which we call synapsis, of meiosis 1 in the first prophase, the sister chromatids of two homologs may actually sort of cross over each other. They make actually physical molecular connections. We call these connections chiasma, or chiasmata for plural, and they're literally places where the DNA of the sister chromatids of two homologs will become connected. And in fact, one piece of one homolog can be disconnected from that homolog and transferred to the other homolog and swapped with the identical piece on the other homolog. Now, we're going to come back to this process later, but keep it in mind, it's a process called crossing over. So one consequence of these homologs coming together in the first prophase is that crossing over occurs. They actually can swap identical stretches, or comparable stretches, I should say, of the DNA on the two different homologs. Now remember, those homologs have the same kinds of genes, so all we're doing is sort of substituting the one parent's version of a gene for the other parent's version of a gene as we swap across chromosomes. And we'll come back to the significance of that actually a number of times over the next couple of lectures. Now, as we proceed into metaphase of the first meiotic division, there's another critical thing that happens. And that is that instead of all the chromosomes lining up in a single plane, the homologs still associated line up in what are essentially two planes, with one homolog on one side of the cell and the other homolog on the other side of the cell. In this case, the spindles are attached to these two homologs separately. Now here's the key thing that happens. When the first meiotic division is completed, what has happened is that the homologous pairs have been separated into the two sides of the daughter cells, or into the daughter cells that are being produced, into the two sides of the cell that's dividing, not the sister chromatids. So as I said before, at the end of the first meiotic division, we now have cells which are haploid. For example, if it was a human cell, there would only be 23 different chromosomes, one of each type, with chromosomes from one parent going to one side and from the other parent going to the other side. The cell has become haploid. Now, what happens in the second meiotic division is actually relatively straightforward because it's as though a haploid cell goes through a normal mitotic division. Again, we have prophase, we have metaphase, and in this case, in metaphase, again, the chromosomes line up. In this case, they line up just the way they did, all the chromosomes along the plane in the center. And then, as we go through anaphase and finish the second cell division, now what happens is the sister chromatids are separated. And what we have now is from one original cell, 4 cells being created. These are the gametes. Each haploid, each having now just an unreplicated chromosome for each chromosome. There's no sister chromatids. And then the way sex works is that these haploid gametes fuse, forming what we call a zygote. And the zygote is, again, a diploid cell. So we reduce by half the number of chromosomes and then bring it back up to the total again. Now, remember that we introduced meiosis as a solution for having chromosome numbers in diploid organisms, okay? But what's really gained by this? I mean, to accomplish this, meiosis has to go through a much larger number of steps, a much more complicated process of moving chromosomes around. This seems like a lot of work. You'd imagine there's something that's worth it for doing all of this. And indeed there is. And the answer is that sexual reproduction, which requires meiosis in this way, generates an enormous amount of genetic variation. Now, some organisms reproduce asexually. In fact, many organisms will reproduce asexually some of the time. But there's a drawback to asexual reproduction, and that is that it produces highly uniform offspring. In In a sense, if you reproduce asexually, you're producing genetic clones. Now, if an organism is well adapted to its environment, and the environment is unchanging, then asexual reproduction might be okay because it ensures that offspring are as nearly identical to their parents as possible, and thus as well adapted to that environment as possible. But more often than not, across evolutionary time, conditions will change. The conditions that offspring find themselves, where they live, the environmental conditions there, will more often than not be a little different from the conditions in which their parents lived. Therefore, there is a selective advantage, there's an evolutionary advantage to producing genetically variable offspring. Essentially, it's a kind of bet hedging. Your offspring will be pretty similar to you, ensuring the continuity of genetic information. They're not just starting over from scratch, but they won't be exactly similar to you, so that they might be able to fit into a broader range of possible environments. Now, where do we get this kind of variation from? We already talked about mutation earlier in the course. Mutation is one process by which genetic variation is introduced into populations of organisms. Sexual reproduction, and specifically the way chromosomes segregate into gametes during meiosis, creates a significant additional source of genetic variation. Let's take a quick look at why this is. Remember, in meiosis, each chromosome behaves independently of its homologue. In mitosis, I should say, each chromosome behaves independently of its homologue. In meiosis, by contrast, the two members of the homologous pairs come together and separate in that first meiotic division. Here's the key point, though. When these homologs separate to the two sides of the cell, the way in which the homologs of different chromosome types line up is completely random with respect to each other. Let me clarify this. Each homologous pair represents one version of a chromosome type received from, say, one parent, the mother, and another version received from the other parent, say, the father. When the first meiotic division occurs, the maternal and the paternal members of different homologous pairs assort randomly with respect to each other as they're segregated into daughter cells, as gametes are produced. Imagine, just to make this clear, an organism that only had two types of chromosomes. We'll call them chromosome type 1 and type 2. That would mean that the total number of chromosomes in the cell would be 4, representing these two homologous pairs. When the first meiotic division occurs, what happens is that the homologous pairs separated, as I said. But these homologous pairs could line up at the first metaphase in two different ways. They could line up so that both the chromosomes inherited from the father originally are on the same side, and both the chromosomes from the mother are on the other side. In which case, when meiosis was complete, the resulting gametes each would have the chromosomes from the same parent in them. In other words, there'll be gametes with just the father's chromosomes, 2 chromosomes, and other gametes with just the mother's 2 chromosomes. But it's equally likely that when the first metaphase occurs, these homologous pairs line up in a different way. For example, the paternal chromosome for chromosome 1 could be on one side with the maternal chromosome for chromosome 2. Whereas on the other side, we'd have the maternal chromosome for chromosome 1 and the paternal version of chromosome 2. What that meant is that when these cells divided, we'd now be producing gametes that had a mixture of the two parents' chromosomes. And in the end, if both of these possibilities occur, then we can imagine there are 4 possible kinds of gametes that could be produced. Now, the interesting thing is not to keep track of all these possibilities. I mean, this is a relatively simple example with just a couple of chromosomes, which is actually many fewer chromosomes than most eukaryotes have. But to realize that what meiosis is doing is actually shuffling the deck. It's shuffling the chromosomes you receive from each of your 2 parents. And in that way, doing something or creating something that we call genetic recombination. And it's genetic recombination, the mixture of genetic material from your 2 parents, that can produce a virtually infinite variety of mixtures of the genetic complements of 2 different parents in the production of gametes. This means that while offspring of sexually reproducing organisms are similar to their parents, thus ensuring the continuity we talked about earlier, they're not identical, thus creating the variation that we discussed. Next time, we're going to look at the consequences of this variation by looking at how offspring inherit traits from their parents in sexually reproducing organisms, following the rules that we now call Mendelian genetics.